And so when you write this in terms of matrix form, so what you say is you have a matrix of a vector of x's that will be related to an adjacent matrix A in this way, and there will be a noise matrix which will somehow propagate this noise through the network. Now, in the stationary state, that means not that this is not dx dt is equal to 0, but dx dt or x will have a distribution, and that distribution eventually will stabilize, and that is the distribution that we are going to be discussing. That distribution will make the elements xi and xj be correlated, and the covariance matrix xi xj will be a function of t, but eventually that function of t will subside, and when that happens, the relation between the adjacency matrix A and the covariance matrix sigma will be this one, which is something called the Lyapunov equation for these systems, and beautifully links the adjacency matrix with the covariance matrix and the noise matrix. So in a way, for us, this is going to be the observable. We can compute from the data the covariance matrix. It's very simply, right? Now, we don't know exactly what is the structure of the noises, but we will have to infer something, and from where we would like to infer A. That will be our inference part.
Can I ask you something? I'm a little confused.
Sí.
The subscripts, they correspond to not to different cells, but to different. So
Yes, I'm talking about one cell and how it evolves.
Okay.
But then,
those fluctuations that you talked about were intracellular, so one cell that has different levels of,
yes,
I don't know, whatever you're looking at different points, so it
Yes.
fluctuates a long time.
yes,
You have the intercellular variability. That's not in this model.
no.
Afterwards, or whatever.
We will have in the second part of the talk for another system, but this is very important point, Silvina. So what I am assuming here is what's happened to this stationary state. This stationary state equation corresponds to what happens in the evolution of one system, which is each cell. But then what we will say is that all the cells are somehow different realizations of this. We will assume some ergodicity here. And so different cells will report on the state of this cell at some other time. And we will make that leap, right? An ergodicity assumption there.
Sorry, but if you are under the linear assumption, why A and Q are not the same?
Q is the noise, right?
Yeah, but
Q is,
so I could have thought that I could have written this equation like xi, the right hand side, as aij xit plus eta or xi of t.
right. Well, remember that there are different ways in which, so what you're asking me is what is the source of the xi? Because if the xi is only coming from if this noise, psi I, only comes from xi, then yes, q and a are the same. But you can imagine, like we said before, there are some things that when we look at, for example, transcriptional abundance, right? Mm-hmm. We are not talking about the number of ribosomes that you have. We are not talking about the number, the structure of the Golgi apparatus. We are not talking about the microRNAs in this case. We are talking about a lot of things that affect the system that are not included in the observables. That intrinsic noise, and to some extent also extrinsic noise, the context, the amount of proteins, the amount of risk or agonode and so on, all that will include things that we are not observing. That is contained in the Xi. Now, the problem with this equation is that if you transpose this equation, it's going to be symmetric. It's the same equation when it is like this and transpose. So, but A, however, has n squared, n being the number of transcripts that we are observing, n squared elements. But sigma is symmetric, so it has n times n plus 1 over 2 elements. And so this equation is not enough to reverse engineer A. And this is the explicit way in which we can say this. The solution for A is basically this equation, where this is the inverse of sigma of the covariance matrix, this is the noise, and any antisymmetric matrix will make this equation true. So we have an indeterminacy based on basically n times n minus 1 over 2 parameters that we cannot determine with this equation. But let's first then think of a system like a physical system where A represents the interactions of a physical system in thermodynamic equilibrium, in which case QQT is basically KBT and the matrix is isotropic, and so A is symmetric, so this U is 0, and so basically what we have here is this equation. That would be the solution. So let's see how it works in some physicals. Yes?
Sorry, so you want to obtain A?
Yeah.
But do you know Q?
We will assume Q. We will assume Q. For example, we can assume, like in this case, that all the elements in Q, except for the diagonal, are 0, and the diagonal is the same as given by temperature. Or you will see in the next equation, we will see that there will be something that has to do with the noise in each transcript, but we will not be able to say exactly the strength. But the qualitative concept of some noise will be enough for the purposes that I have for this work. But yeah, we don't know it. It would be.
I mean, like in the future, Q is something that you think you should know because you can extract from the experiment. That's the idea.
In principle, yes. In principle, yes. Okay, so for example, suppose that you have this simple system of a chain of springs, and each particle has the same mass, and we have a spring constant and some noise in each of them. So each noise is going to be independent in each particle, and so this is the set of equations. And so the covariance matrix, you can infer from, simply compute the covariance matrix from this in this case, based on this, K is don't confuse K Boltzmann with the strength of the spring, M gamma, so gamma is the viscosity. So it's a fully connected, it's a full matrix, right? There is no zeros here. Which is interesting, right? Because what we were saying before is that when we typically do reverse engineering using the simple concept that one gene has to be correlated with the other in order to be, and then we will infer that they are connected. Well, we'll see here that the covariance, the noise will propagate through all this chain, and so the covariance between 1 and n is going to be different from 0. However, if we use our formula, right, we said that A, the adjacency matrix, telling us the interaction between the different entities is simply the sigma to the minus 1, we have exactly, obviously, because the equations are the ones that we were working, exactly recapitulate the interaction matrix, which is very interesting, right? Is the inverse of the covariance that recapitulates the interaction between the elements, and not the covariance matrix itself, even though conceptually is the covariance matrix, the susceptibilities between the things, that will tell you something about the interaction between things. In a slightly more interesting, because it's not coming from the same exact theory, we can assume that you have a spin model with some magnetic field and interaction J between the spins, and then from that system you can compute the probability of a state, and then you can compute the covariance between the states. Here states are spins up or down, which is simply this. This is the covariance matrix of spin I and spin J. And again, it's a full matrix, right? However, the interaction A, if we use the same formula, KBT times the inverse covariance, we have that only nearest neighbors interact. And if you, R is this thing, but if you assume that J is much smaller than KT, then this R becomes, this interaction matrix gives you exactly J in the nearest neighbors interaction. So the theory, even though it's not created for a spin model like this, still recapitulates the interaction. So so the leap of faith here is that there are some assumptions that are not exactly validated. For example, the ergodicity, or for example, the stationarity of the state. We will assume, for example, that the processes are such that systems are in quasi-equilibrium even though they are still evolving. Okay, so let's first, so how do we use this in this case? So we'll use it in the most simple case, in which we will assume that we have a two entity system. Each entity will have its own noise, and there is some interaction between 1 and 2, but going from 1 to 2, and not vice versa. So in this case, A would be the degradation of element 1 and 2, and one of the two extra diagonal here will be non-zero, given by the directionality. The noise will be assumed to be some strength of noise in each of them, and Q1, Q2 we don't know, so we will assume it's something. We also don't know K. And remember that this is in the log scale, so we are trying to say the log of 1 will affect the log of 2 in some way, and we will assume that K is this local response. Now, if I use this equation, what this tells me is that sigma, the covariance matrix, I just use the unknown A and the unknown Q to pretend that I can solve for sigma here, because now, and what results here is that a structure for sigma that looks like this. Some element here in 1, 1, some element here in 2, 2, this is the variance of 2, and you see that the variance of 2 depends on the covariance of 1 and 2, which you can get from this element. However, the variance of 1 doesn't depend on anything. It's exactly what we were saying. The noise of the first thing will affect the noise of the second through the covariance here. And there is a specific prediction here, is that the variance of the second element, the downstream element, varies quadratically with the covariance between element 1 and 2.
Sorry, the invariance that you mentioned before, you don't have it here. The arbitrary U matrix that you mentioned.
No, in this case, no, because remember that in that case, what we are saying is that if I have sigma and Q, I cannot solve for A. But here I say, I know A. I know A. What does it imply for the structure of the covariance matrix? And from that structure, I can infer that the variance of this downstream element depends quadratically on the covariance between 1 and 2, but the variance of the first element is independent of that covariance. So somehow we are thinking of it differently. Now, yeah, but then I'm trying to go back and say, okay, if my assumption about A is true, that K is only 1 to 2 and not 2 to 1, then what does it tell? And then I will be able to do a test. Okay, is K in the 1, 2 or in the 2, 1? Because that's what I want to say. Is MEC phosphorylating ERK or ERK phosphorylating MERK? That I don't know. So in that sense, I will address the inverse problem.